All right, I think we're gonna get started today. So hello, my name is Bernie Lee. Welcome to this AI ready data with Biologics LIMS. Really appreciate you being here. My name is Bernie Lee. I'm a product manager here at LabKey Software. I plan on briefly talking about some of the data management challenges of biologics discovery and development, and the tools and strategies that we employ to drive success, to improve efficiency and just generally increase the quality of life for our scientists. And then I'd like to spend most of the time today actually stepping into the product and giving a glimpse of some key features. By the way, please ask questions. There's a Q&A and chat feature of this Zoom, and at the end, at least, I'll take some time to answer any questions that are there. Thanks so much. So we are going to be exploring these ideas through the lens of our biologics LIMS, which briefly can be kind of a one-stop shop for a lot of scientific endeavors. But for some more established information architectures, sometimes bigger companies and institutions, it can be a great hub for centralizing data from many sources. So our product was originally designed for antibody discovery and development, but supports a wide variety of other biologics endeavors today. It provides for accurate tracking of entities like sequences of molecules that you'll be commonly working with, as well as samples, plates, and assays run on them. And it helps bring data together for analysis, reporting, decision making, project planning, all of that kind of good stuff. It does so through a series of feature areas, places for sample management and assay data management, areas for analysis, reporting, visualizing a lot of that data, a place to register and track and enrich information about biological entity registration. I mentioned sequences of molecules, but it includes anything else that you're interested in, your cell lines, vectors that you might purchase and use, all kinds of other entities. And I also mentioned plate management before, the ability to track iteratively through time, through operations, what plates you have, what samples are on them, what's derived from what, and to roll data up and down between those in an easy way. A way of automating a lot of workflows, and our data connected ELN, which allows you to comprehensively document everything that you're doing in one cohesive narrative. So there are a few big challenges that I'm going to mention briefly today when it comes to managing data for sophisticated scientific endeavors. The first one is lost and confusing data. People lose track of samples, like the physical samples, but they also lose track of data associated with them, or they can't make use of data after the fact because it's stored in some misnamed spreadsheet or a PowerPoint somewhere, or it's in a standalone ELN in another piece of software that you can't find, forgot about, or isn't useful once you've written it. There are also some limits to post hoc utility, given that discovery and development often require multiple iterative rounds of work as well as handoffs between scientists. It can be particularly difficult to track data as it goes back and forth and through all of these things. The example that I like to use, that I like to give, is going from initial primary plates, from which you are pulling material in order to screen it or do other sorts of sample preps, manipulations, and then ultimately being able to take that downstream data you gather and have it on that original set so you can properly pick the materials that you want to continue to work with. It can be a very painful spreadsheet game if that's the way you go. And then the last one that I wanted to mention was just the loss of time. And this one's really big. Loss of time and energy, motivation for scientists. These challenges can cause scientists to have to redo a lot of experiments or spend a lot of time digging through their computer or file stores somewhere, looking for old data to try to reconstruct analyses and reports. What we really want is to ensure reproducibility across multiple assay and experiment setups, across different scientists, across different teams, sometimes across different states and countries, across projects, and it can be a lot of time, at least hours, days, weeks, sometimes months to reproduce some of this critical data. And in the data science era, this one where everyone's trying to figure out how to take advantage of machine learning algorithms and AI applications in order to speed up their science, draw novel and additional insights. Those applications are very heavily impacted by a lot of the same challenges that we're talking about. So whether it hurts you and your scientists today, or it impacts, slows, or entirely hinders your efforts to make use of some of these advanced data science applications, it's a problem. So what makes data AI ready? Well, it depends on the application, certainly. Large language models like your favorite chatbot can consume and probably interpret your scientists' notebooks, if anyone can, but they're going to have more trouble with well-structured data. So there are things like deep learning models, neural networks, things like digital twins that they can't consume your unstructured notebooks so well, but they can take structured data about your sequences and about your molecules in association with a lot of the downstream assessments that you're doing of performance, either to choose what candidates to advance in your R&D process, or in order to choose the ones that are going to ultimately be successful in clinic, and can use that tightly related structured data to ultimately predict outcomes for you. And then they can reduce the need to run a lot of costly confirmation experiments, and obviate the need for things like excessive production runs. All of that's very appealing. A lot of those things are the most costly in terms of time and money, aside from the clinical trials, of course. But getting there can sometimes be kind of a big challenge. But it's very useful, among many other things, but to name a few, to have structured data, richly annotated with metadata, related to and captured alongside your experimental results. So that means your data is organized and defined. It has standard identifiers. It has, again, rich metadata that describes what the information is that you have captured and are viewing and exploring. And doing so only some of the time is going to leave you with huge gaps, either for your people, like I mentioned before, or for these machines. So you want to consistently do predictable data capture and consistently use things like the same data shapes and the same naming conventions for individual scientists who do this stuff regularly, but also across teams, across projects, across the whole company, the whole endeavor. And then the last one I'll mention here is that you want to use interoperable formats and accessible tools so that all your work doesn't require a doubling of effort just to make use of it later. This is kind of what I was talking about at the very beginning, where if you shove in an unstructured way all of your information into some ELN somewhere, or you have it in files scattered all over the place, you then have to go through a whole bunch of work to take that and put it in a form which is then usable for some kind of broad-based analysis or machine learning algorithm. Okay, with that, I wanna show you a little bit of the product. So everything that I said there, all fine and good. Maybe it sounds a little oversimplified, but that's what follow-up conversations are for. So after this, we'll send you all out a link so that you can consider having a follow-up conversation with us about some of these things. Talk to us about your use cases, and let's see if we can expand upon what I'm talking about here today in a way that's useful to you. So again, now I want to show you some of the aspects of our biologics LIMS. how it's oriented towards artificial intelligence applications and machine learning applications, with an open invitation to set up some time with us later to do a deeper dive. All right, let's jump in. All right, so here's the application home screen. You can see a bunch of basic insights about what's in the application, information about your existing samples and where they live, things about sample management, storage, jobs list, existing notebooks that are out there. I'm actually going to start us in a fake notebook that I took from an existing manuscript publication that was about poxvirus display antibody selection. This is a really basic notebook. It has a series of sections, an introduction, materials and methods section. It has an area with results, charts, data, tables, all of discussion at the very end. All of this is fine and good. It allows you to capture all the information about the narrative of the scientific endeavor that you and perhaps others are participating in. We support co-authoring on these things, so if it's a multi-person process that's involved, everyone can jump on here and make their contributions. You'll see that we have these little pips on here that relate to, in this case, a cell line, 2 cell lines. Here's a molecule. These notebooks, while comprehensively written up as text, as data, as images, also are aware of the data registered in the system that it's associated with. So this is actually a reference to a real entity in the system, which I can click into. Here's a molecule, gemtuzumab, in our system designated as M2, molecule 2. And because the notebooks understand all of the data that they're associated with, the molecule itself, its downstream samples and assays, which I'll show you this in a second. If you were to feed your notebooks into a large language model, it could understand the associations between your write-up and the actual data that you have present, in order to draw faster insights or help you author future notebooks. There are all kinds of basic applications here. Okay, so this is a virtual entity. It's a real molecule with a structure format, calculated physical properties. You can see component protein sequences that build this thing up. I can come in here and you can actually come and see the all the annotations on the sequence itself, and understand again, average masses and isoelectric points, as well as associated downstream samples that contain this thing. I can not only see that I have some processed samples, I have some protein batches, I have some stability samples that for formulation experiments on these things, but any assays run on samples of this molecule, we can see all aggregated together. So here's an amino acid composition for this molecule. You can see some cell viability data for the cell lines that have produced this thing. You can see some residual DNA data, some titer data. So all of this stuff is in reality run on the samples that are associated with this molecule. We roll it up all together for you in a structured way, so that if you wanted to see, for some particular signal peptide, when you use it for your IgGs or some other particular structure format, how does your titer come out? or some other kind of downstream assessment like that, those relationships are formed and maintained in our system in a structured way, and you will be able to, as you build up, as you aggregate data on such things, you'll be able to use things like machine learning applications to do some of those predictions. Okay. Oh, I should show you these relationships. We produce visual diagrams of a lot of these relationships. So here you can see I'm looking at an expression system, and a couple of the batches of protein produced. You can see all of the contributing protein sequences to this molecule. Here's a different view of that. So not only do you get it in a structured way so you can make use of it for computer applications, we give you this nice view of the relationships of your data for use elsewhere. Okay. We can take these protein batches and we can act upon them. So I have a set of 5 expression runs, protein batches in the system, and I wanted to briefly show you how you can manage these samples as well as make use of them in your experiments. So you can see that these expression runs are associated with various molecules in the system. You can see quantities. You can see that they're in storage. I can actually click into here and see this thing in a box or in a plate on a rack in a freezer. I can check these things in and out, track freeze thaw counts. All of these things are great and useful. I can generate aliquots of these things. I can add them to jobs and request other people to do work on them. I can view reports on these materials. But for now, what I want to do is take some of these materials and actually put them on a plate and run an assay on them. So in this case, I'm creating a demo fusion plate, and I'm going to spread this across 4 plates. Oops, why did I do that? Mistakes are made. I want to generate dilution curves across multiple plates, and I've pre-created a set of templates, which I'll show you in a second, so that I can simply select these samples, throw them on the plates, and generate fairly sophisticated plates for my experiments. So it created this demo fusion plate, and because I had a pre-configured template, it automatically took these samples that I was looking at, these expression runs 1 through 5, these protein batches, and arrayed them as dictated on this plate. So you can see that each of these come in, when I'm looking at these 5 particular wells, it shows me basic information about them. The outer rim of these plates are all blank. If you go along these rows, you can see that it's this dilution curve. And the same thing is true across some of these other plates. It's a lot of replicates of the same curve, so that when we gather data against these, we can generate curve fits and EC50 values and things like that. So once I have formed up these plates, it's as easy as saying I want to import data against these, and then I can drop my plate read right here. I'm actually going to just show you that. So I have an example plate read here. This is a multi-plate read, with very low values around the outside of those blanks. And as my dilution curves increase in concentration, I get darker and darker wells. So again, I can just take this and I can drop it in here. You should show up in a second. There we go. And simply hit import. And what will happen is that LabKey will consume that plate read, and it will take the values from the spreadsheet and automatically associate it with the sample in the well in that plate in your folder. Okay, that's automatically done for you. You don't have to do some manipulations to rearrange things or copy between one spreadsheet and the next. And because we do this automatically, and because these samples are related to those molecules, you get that automatic roll up. So you can see for a particular molecule, what these, in this case, fusion protein interactions look like. Once the data is in, obviously I just brought in the raw data, but because these are replicates, we automatically calculated some means and standard deviations for you, which you can then use to create some charts. Here are those curve fits on a pre-created chart. So simply by taking the samples that you're working with, plating them, and throwing your data at them, you get a bunch of free analysis and reporting. And you can look at the various statistics associated with this. And again, that data rolls up onto those plates, for which you can do, if it's appropriate, some kind of hit selection, or reporting, or just decision making about what you ought to do next. And all of this stuff, the assay data, the plates, the samples, the molecules, can all be in that notebook in a comprehensive way, so that you have a one-stop shop for re-exploring the data and work that you've done. Okay, I wanted to keep that kind of simple. We are going to jump back over very briefly to these slides for a little closing and Q&A. So fundamentally, the cost of designing or discovering and then developing a biologic is largely driven by the amount of energy required by the people that are involved in the instruments. And I just showed you a few aspects of our biologics LIMS, which can reduce a lot of that energy for an institution, driving efficiency gains, being organized, having all that information in one place, a lot of the automatic data association, a lot of the automatic data analysis. So by using a tool like this, you can seriously reduce your dependency on spreadsheets. You can reduce your dependency on humans, which inevitably reduces errors, and tends to speed things up. Also reducing the time required to track and report on your operations. You can use our system to track and maintain uniqueness of various entities. So I showed you one molecule in one sequence, but of course you will have somewhere between dozens and thousands or tens of thousands of those entities, and making sure that you aren't accidentally registering the same one, like a dozen times under different names, and then you have to figure out how to reconcile that data is really important. So we help you do that, reducing the amount of reconciliation and repeated experiments may be required in the future. I showed you how to automatically capture and analyze a lot of that data and form up those associations. We try to keep that simple and easy. And I very briefly showed you our data connected ELN, where you can document and report on your findings, as well as share those things both internally and externally for your collaborators. And what you end up with are many fewer regenerated reports, happier people in your bioinformatics and data science group, happier managers, happier C-level and VP level personnel, and just a more efficient institution in general. So ultimately, you can reduce that energy, reduce the costs by imposing some order on your data through LabKey. So I want to thank you very much for joining us today for this quick tour of our biologics LIMS. I'd really like to take a deeper dive with you and learn about your use cases, your pain points, your goals. So keep an eye out for an email from me that has a link where you can schedule a time we could talk a little bit more, could do a little bit of a more tailored demo to your organization.